Hi. I'm Yuki Naitou from Technical University of Dresden. We know that lipids are really essential molecules in our body, and they are so diverse. For example, in eukaryotic cells, there are more than 2,000 lipid species. So the big question in my project is, what is the physiological function of this lipid diversity? So then I focus on the small intestine because most of the dietary lipids are absorbed in the enterocyte in the small intestinal cells. We can easily imagine the variety of the dietary lipids. For example, fish oil, meat fat, or butters have different health impact, right? But we still don't know how these different lipids are actually processed in the enterocyte. So I aim to visualize this lipid processing in the enterocyte using bifunctional lipid probes. So they are photo-crosslinkable and clickable, allowing us to trace the lipid transport in the cells. So what I do is parse-trace experiments. So I load this lipid from the apical side in the cell layer, like this, and and after a certain amount of time, those lipids are transported into the internal organelles. And by fixing those cells at each time points, we can visualize and trace the lipids behavior in the cells. And interestingly, I found that the lipid transport from apical membrane to ER is super rapid. At even 1 minute pulse of the lipid loading, those lipids are already internalized, as you can see here. And this super rapid lipid transport can be slowed down at the lower temperature. And by combining the organelle staining and the segmentation, I can quantify the for example, phospholipid transport, showing a decrease in the apical membrane and the increase in the ER. So far, I have established the lipid imaging pipeline in the enterocyte, and also I showed the lipid transport is super rapid and also temperature dependent. So if you're interested in, please visit my poster number 30. I would be happy to discuss more. Thank you.